Hello, everyone, and welcome to phyloseminar.org. The current theme is single-cell analysis, and this is the third talk in a series of 3 talks on that topic. Please use the YouTube live comment box to ask questions. Today's speaker is Dr. Sophie Seidel. Sophie did her undergraduate studies in molecular medicine at the University of Tübingen, where she worked on the proteogenic genic potential of non-coding regions with André Lupas. She then completed a master's degree in computational biology and bioinformatics at ETH Zurich, focusing on systems biology and phylogenetics. Sophie is currently pursuing her PhD with Tanja Stadler at ETH Zurich, where she develops phylogenetic methods for developmental biology applications. Sophie is broadly interested in techniques to trace cell lineages and to model the resulting data to learn more about the development of organisms and tissues. Welcome, Sophie, and thank you for participating.
Yeah, thank you so much, Eric, for giving me the opportunity to present here and to show our recent work. Well, everything I'll show here today is now published. We are kind of still working on extending a lot of these concepts, and there are still a lot of challenges ahead. So I'm really grateful for any input, comments, and questions you might have. So feel free to interrupt me at any moment in time. Exactly. So as Reza Kalhor and Sebastian Prio already motivated in their previous talks, I'm also interested in lineage tracing data in combination with single-cell omic data. And just to bring everybody here on the same page again, so in lineage tracing, we have some kind of synthetic genomic construct that's introduced into a tissue or an organism, and that allows us to record some phylogenetic signal and for single cells. And this is nowadays combined with various omics technologies, for example, single-cell RNA-seq, but you could also imagine methods to identify the availability of certain promoter regions. So you've got a variety of different readouts that allow you to identify cell types. And using both of these joint or measurements then allows us to really reconstruct lineage trees that we can then annotate with, for example, the cell type information at the tips of the tree, but also using other techniques could allow us to even know more about certain situations at the internal nodes. So this is really an exciting new field, both on the methods side, where we, as kind of the phylogenetics or methods development community, are really challenged to think how we can adapt our methods to this new data source and the assumptions, but also on the biological side, where really still a lot of development is happening, and it's quite exciting to see the experimental community come up with an entirely new thing that they can measure almost every other year. And this lineage data really just allows us to answer a whole new set of questions that I think is pretty much inaccessible from these other omic measurements alone. And so one of these questions one might ask, and that that was kind of the starting point when we were thinking about this project, was let's say this pictorial example here was really the lineage tree of any model organism, and you would have single-cell RNA-seq, so gene expression data measured at different time points. These would be indicated by these gray bars here. And this would very much give you, then you would know how many cells of each type are present at these time points. But what you're really lacking if you don't know the lineage tree is what kind of cell dynamics lead to these shifts in the cell plate frequencies, whether cells divide more, die more, or how they differentiate into each other. And so what we really, what our major goal is really to understand like these cell dynamics and to make quantitative statements about these to narrow down what a state of healthy development is in a quantitative sense, and then ultimately to also identify what processes are then deregulated and if development goes wrong. Yes, and so we try to do this using phylodynamic methods, and as this audience is probably very well aware of, phylodynamic methods generally are methods to quantify population processes based on a phylogeny. And to just give some brief examples of how these methods have been used in the past. Here on the left, I show you a species phylogeny of bears. This was used to estimate the speciation and extinction dynamics over millions of years. And on the right, you can see more of a pictorial example that should give you an impression of how a phylogeny might be used to assess the mixing of types or how to measure the population structure very much. So here on the right, types would change very frequently from one to the other. Meanwhile, on the left, types are more preserved. And when we think of bringing these methods then into this single cell biology / developmental context, we would really like to quantify then developmental processes in terms of the following parameters. So we would ideally want to have type-dependent parameters. So certain cell types should have different, should be able to have different parameters, and these could be cell division rates, apoptosis rates, and differentiation rates, but at the same time accounting for sampling. However, when we started out with this project, there weren't really methods available to estimate time-scaled phylogenies from lineage tracing data. And that's where we started. Because to estimate these parameters in a phylodynamic framework, this is really what we needed. Exactly, and this brings me to really kind of, the goal of this talk, to show you our method TiDeTree and how we use it to infer time-scaled trees. So I'll quickly introduce now the data that, in a more formal way, that we will use in the following. So we assume that every cell, especially the starting cell, has a barcode, so a genetic construct that's introduced into its genome. And it might have several of those, and these barcodes are scattered through the genome, so we can assume that they are independent. Then an enzyme edits this barcode irreversibly at some point in time. And in the end, we expect that cells with similar barcodes should have a shared ancestry. However, there are a couple of challenges that are involved with this data source. One of them is that some of the editing outcomes of these indels are recurrent. And so most of these enzymes are biased towards one particular editing outcome. And what a lot of methods previously did is to remove the data from these most frequent indels in order to avoid biased inference. However, we would really like to use them because they contain information nevertheless. Another difficulty is that the editing time, so the duration during which this enzyme is kind of active, is usually a lot smaller than the entire developmental time. And this has some interesting challenges on the signal that is contained in a phylogeny. And then also we would not only like to infer the timescale tree, but also the cell dynamics from it. Right. So in order to do this, we developed this method TiDeTree, and TiDeTree really assumes that there is an unobserved population process that underlies the data generation. And I've kind of, so this is maybe a lot of the plot at the same time, but I'll walk you through this slowly. So at the start of the process, you have a starting cell with a barcode. So this is then here at the same time depicted as the number of cells in yellow, and you start at 1. And then as the cell divides, you get 2 lineages, and then your number of cells reaches up to 2. And so this number of cells very much always tracks is kind of a summary statistic of your population process that goes on in the background, and that is captured by the full unobserved phylogeny. However, we usually only have data from a subset of cells. So in this case, these would be these 3 black tips of the tree for which we have actually sampled the sequences. And for those, we wish to reconstruct the true phylogeny. And we also want to estimate this unobserved population process based on this simple sample. Exactly. And so as standard practice in phylogenetics, we now take this input very much here and would plug it into a tree likelihood formulation and would like then to estimate a timescale tree under our editing model. And this editing model is something we have developed for this particular type of data, and I will walk you through those in detail now. So we decided to model the editing process explicitly. So here on the left, you would see, again, the full unobserved tree, and on the right, the sample tree. And then you can observe different states on those. One is the unedited state that we assume the starting cell always starts in. Then cells can, any barcode can be edited, so transition to 1, or it can be silenced. So let's first talk about editing. So editing can happen in what we call the editing window. This is kind of this green, greened out, green shaded region here. And currently in most lineage recorder settings these days, T1 is either exactly 0 because experimental biologists would inject the editing enzyme into the starting cell, and then you would have editing practically starting right away, or they would only induce the expression of this enzyme at a later time point. So it makes sense to actually have T1 as a parameter that can be varied, and then T2 is usually, so the end of this editing period is usually determined by some independent experiment where one can observe that the editing period saturates. Right. Okay. So that's about editing. Silencing, we assume, can occur throughout the entire time period. And silencing in these contexts means that a genomic region is compacted so that its DNA cannot be read off any longer. And so we wouldn't observe any RNA transcripts. And this is the readout most of the time, single RNA-seq. And so we might not be able to observe a transcript just because it was silenced during development. Okay, so how do we translate this into our model? We have yes, we have an edit rate, or what is usually known as the clock rate in phylogenetics. That is the rate at which any edit is introduced. And then we also model, given that an edit is introduced, a probability distribution over the different indels that can occur. And and then we also, in addition, have a rate of silencing. This is rate L, at which any barcode can be silenced, no matter whether it is unedited or has been already edited. And so, yeah. So, and is that if you set these parameters up in a transition rate matrix, then it turns out that the transition probability matrix can be calculated analytically, and this makes actually this calculation fast no matter how many. So that the dimension of this matrix increases for the number of editing outcomes, and yes, and so yeah, we can compute these transition probability matrix in linear time.
See, before you go on, so do we observe silencing directly using something like ChIP-seq, or is it sort of like it might be silenced?
Yes, that's exactly right. We don't know that it is silencing. Exactly. So this is an assumption we would make. Yes.
So essentially there's some latent state, which is like, is it silenced or not? We don't observe it, but if we don't observe any reads from that region, we say it could have been silenced.
Exactly. And I mean, I think this is a very important point to make. I think this will also be one of our first extensions, is to also introduce then an error model, such that we would be able to distinguish, or distinguish, but I guess co-estimate whether, because you could also not observe a read because you just didn't observe it during single-cell RNA-seq experiment. Exactly, but we don't model this currently.
And right, does it ever happen that you sort of like, there's so much editing that these aren't primed, or wait, are they? Are you using primers? Yeah, you're using primers to amplify these regions. No.
Right. Yes, they do. They would do targeted sequencing on those particular regions.
So would it be conceivable that you would have so much editing that you just wouldn't be able to amplify a certain region?
I don't think so. Or I would hope not, because usually, well, I guess you could have the situation that you cut out so much that you would also, I guess, cut up the primer that's used to amplify that region. I think in the data sets that I've currently worked with, we have more had the problem that there wasn't enough editing going on. Yeah. And maybe also something on the question you asked before, on the silencing. So something that we did observe, so this is a project in brain organoids, is that the longer kind of you wait, so the longer you allow your organoids to grow, the less of the transcripts we would observe in the sequencing readout. And so that led us to think, okay, there's also so it's not only sequencing dropout that you would expect to be the same whenever you sequence, but there's also a temporal component that the longer the organoids have time to grow, the less of these transcripts we observe. And that's what led us also to include this silencing. So it's not directly ChIP-seq, but there was this temporal component as well. Yeah.
But do you assume that all the root state is always like an unsilenced state?
Yes. Yeah. But I guess we would only have the root state if it is observed in any of the cells. And so, well, I guess then we're also making this assumption that you cannot unsilence. But yeah.
Okay. Thanks.
Yeah. No, but yeah, thanks for asking. These are certainly things to keep in mind. Yes, so kind of given that we have this in the distribution, we are able to account for our recurrent editing outcomes. And also we now explicitly have a formulation that it explicitly takes into account that we have only limited signal during this editing time period, and then less for the maybe remaining developmental time. But how do we move on to the cell dynamics? So that was not so much work maybe on our side. So we just implemented this likelihood calculation into the Beast2 platform, which generally supports Bayesian phylogenetic analyses by sampling those trees. And so, within these two, there are a lot of phylodynamic models available that can estimate some population processes based on a timescaled phylogeny. And we use those to parameterize our developmental processes. Exactly, so that's how TiDeTree, it's our attempt at addressing these challenges. Great. So
maybe I can. Okay, I thought maybe maybe you'll answer this question later, but I'm not sure that you will. So like when you're doing your MCMC, like a single state includes everything that is not shaded in here. So it's sort of like a tree on the observed sequences and also a population size trajectory. Is that correct?
Yes, and potentially the editing model parameters.
Right.
Yeah.
So and the point is that by having the number of cells there, like, is there also sort of, is it latently also sampling these dead lineages?
No, it's not.
But so
I mean,
the point is that if you have this population size trajectory and you see that, for instance, there's 4 more cells than you have in your observed sample at a given time, that allows you to calculate a likelihood that, like the
oh, sorry,
full
I think I didn't say this correctly. So we don't sample these trajectories. We only sample the trees.
Okay.
So we very much sample the trees, the editing model parameters, and the phylodynamic parameters. So, and you can calculate the phylodynamic likelihood without having to take into account these population trajectories.
Okay, so what role does the population trajectory take here?
Oh, this was just more kind of to illustrate that there is kind of an underlying population process, and that we don't observe kind of the full reality, and that we kind of have to make our inferences based on a subset.
So the point is that this is quite nice. So the point is that if you have an observed tree and a model of phylodynamic likelihood, you can calculate the likelihood marginalizing out all of the possible
trees.
See, we're looking at a single observed tree. I'm thinking about, are you marginalizing out all the sort of hidden state, like all these population trajectories and things like that? Like there's in this picture, there's a lot of hidden state. And I mean, if you have all of that hidden state, all of these dead lineages, then it's relatively easy, I think, to calculate a likelihood, like the full phylodynamic likelihood. But marginalizing, I mean, this is sort of a persistent problem in phylodynamics, that you have these extinct lineages, and like how you calculate the likelihood in the present, like if you're not explicitly having the extinct lineages as part of your state.
Right. I guess this is kind of taken into account by the birth death sampling model, right? Or like whatever, well, by your phylodynamic model, at least how I currently understood them. So if I provide, so under my tree, or like the probability of observing these population dynamic parameters given my observed tree, already take into account that I have that all my lineages have some probability of going extinct at some point in time, right?
Yeah.
So I mean, or maybe I'm not understanding your question correctly. So I thought,
I mean, we would like to be able to calculate the likelihood of this black tree, the observed tree, under a phylodynamic, like a complete phylodynamic model. In my experience, it's challenging to calculate the likelihood of just the observed tree without saying anything about what these ancestral, like these extinct lineages were doing. And so, for instance, like there's this multi-type birth-death process.
Yeah.
And like the main content of that paper is like thinking about how we can calculate these likelihoods under an approximation, how we can calculate these likelihoods. And one of the things that they do is to have an approximation based on like no changing in extinct lineages. So do you leverage any of that, or am I totally off base?
Right. So So I think, so for this, maybe two things. So first of all, so we only make here use of the existing implementations of these phylodynamic models that are already present in these. For the remainder of what I show, we'll just work with a homogeneous birth-death model, so not multi-type.
Okay.
And there you don't have to track these extinction probabilities, or like, I think you can really just, there's an analytical formulation for calculating the likelihood of your population dynamic parameters given your observed phylogeny. But I think you're right for the multi-type model. I think there they have these differential equations that they calculate, I think, from the tips all the way up to the tree, and they have to do some approximations there.
Perfect. Yeah, this totally answers my question. And
okay,
I get it. Thank you.
Okay, cool. Yeah. All right. Exactly. So let's move on to some results. I first will want to show you some model validations on simulated data, where we looked a bit into, is the model correctly implemented, and can we extract signal from the data? What happens if we have more data available? And then I'll also show the model on some experimental data as part of a benchmark, and then also trying to infer some simple cell dynamics. Right. So first, to check that a model is correctly implemented, we perform what's called a well-calibrated analysis. So we first define a prior distribution. So we just specify some distribution over all our simulation parameters. Then we set, that's fixed for all simulations, that editing occurs only for half of the experiment time. And then for each simulation parameter that we've drawn, or set of simulation parameters that we have drawn, we simulate a tree under both the sampling model and an alignment under the substitution model that we implemented in TiDeTree. And then we perform the inference using TiDeTree. And then the general idea is that we would ideally like to have what we simulated, the true simulation parameters back. And in these well-calibrated analyses, what we usually compute is the coverage. This is the, we very much calculate how often does our true simulation parameter fall within the 95% highest posterior density interval. And this should happen if everything's well calibrated 95% of the time, based on how this interval is defined. And I show you now the results here for 4 parameters of interest. The tree height. So this is the time between our samples and the most recent common ancestor. The tree length, which is the sum of our branch lengths. The birth rate, which is the parameter of our population model, and would in developmental context correspond to the cell division rate, and the sampling proportion. And all of those converge very much to 95%. So this indicates correctness of our implementation. We then further wanted to look into how much signal can we actually extract from this data. And so here I kind of split our kind of data sets into those that have phylogenetic signal and those that don't. So what does having phylogenetic signal mean? So phylogenetic signal means that your tree is, that your tree root, so your most recent common ancestor, is within this editing window. So this would, for example, hold for the MRCA of these three samples. You can see this would be the MRCA, and then the sequences have time to acquire some different mutations. So we actually have signal to reconstruct some phylogeny at all. However, if the root of the tree is after the editing window, so this would, for example, be the case if we were only looking at these 2 tips, the MRCA is here. This bar, these cells don't have any chance of acquiring mutations that make them different. And so these subtrees will not contain any phylogenetic signal at all. And so when we then look at the correlation for these, segregating for these 2 types of data sets, we can see that for the tree height, the tree length, and the birth rate, we can essentially infer the parameters a lot better if there is phylogenetic signal, as we would expect. However, for the sampling proportion, there doesn't seem to be a difference. And it also seems to be the case that the correlation is a lot lower. So we are currently investigating whether this really means that we cannot estimate the sampling proportion, or whether, and at least based on, these data, we didn't want to make a conclusive statement, but it seems to definitely be the case that the birth rate can be estimated a lot better. Okay, so then we're interested in what happens if we have additional data available, because this is really a case that happens very often in experimental biology. People don't only have a single alignment, but they most often have a single alignment and and they actually do know what indels occur at what frequency because they have some independent measurements, scenario B. And most often they would actually have experimental replicates. So they might perform the exact same experiment multiple times. And so you could imagine having, I don't know, alignment 1 from fish 1, alignment 2 from fish 2, etc. And then what we can actually do within TiDeTree is use all these input alignments within the same inference and then pool the population process parameters across them, or assume that they all follow the same population process. Exactly. And so we then looked what happens to the inference results under different metrics. And I'm showing this just for 2 metrics here for the sake of time. This is the highest posterior density interval. So kind of can very much think of this as a posterior interval. And the larger this is, the more uncertain we are about the precise value of the parameter. So here, the lower it's better. And then the RMSE is the root mean squared error. And this gives us a measure of accuracy. So also here, lower is better. So for the tree parameters, so for the tree height and the tree length, we can see that adding the indel distribution improves mostly the accuracy, and also adding experimental replicates also improves both of the parameters in a minor way. And then for the population dynamic parameters, we see that the precision is mostly improved by adding experimental replicates, and then the accuracy is improved by both adding the indel distribution and also experimental replicates, which follows our expectation that additional data should also improve our parameter estimates. Great. So let's now move on to some experimental data. And we used a public data set that was available as part of a dream challenge. So which was really a community effort where different methods were benchmarked on this data set. And the data set is collected as follows. It has 10 sites. And then each of these 10 sites can be edited by an integrase. And then this integrase basically leads to do different editing outcomes, either deletion or inversion. So the space of different editing outcomes is really not so large. But what is really nice here is that these cells are grown in culture and they were imaged throughout the entire time period. So we can actually, from this movie, really know the ground truth tree topology and even have some estimates of the true cell division times. And so they generated 106 data sets with this and split them within this challenge into training and test data sets for methods to be trained upon. And so the general idea is you get these alignments, then methods should kind of infer a tree, and then you can compare this inferred tree to the true tree that you have available independently. Right. And so this benchmark worked on to evaluate the tree topology, 2 different metrics were used. One of them is the Robinson-Folds metric. So this really asks which bifurcations that are available in the ground truth tree are also in the reconstructed tree. So here's an example. This bifurcation or like the cell division event that leads to A and B is present in the ground truth tree, but is not in the reconstructed tree. Compared to that, this bifurcation leading to EDC is also present in the reconstructed tree. And then you compute the ratio of those, and this gives you Robinson folds distance. Compared to that, the triplets distance is basically in a similar spirit. So you're very much asking which of the 3 taxon kind of subtrees that you can extract from your true tree are also present in the reconstructed tree. So here really A, B, and C, you would kind of extract from the ground truth tree and then compare whether they are also present in the reconstructed tree. Right. So these would be the two tree topology metrics. And we then use these to rank all of these methods with respect to both. And here also, the smaller the value, the better. And you can observe that, so here would be TiDeTree, and here would be Ambivalent and Cassiopeia, which was the method that was shown by Sebastian Prio. So TiDeTree really performs comparably in terms of tree topology estimation. And what I should also say is that TiDeTree doesn't make use of the true training trees at all. We really just used all available alignments and ran TiDeTree on those. And I think something that I should also mention is, so here we just use the maximum a posteriori tree. But in general, TiDeTree infers the whole posterior distribution of tree. Meanwhile, all of the other methods just infer a single best tree. And I'd just like to make, like to show how maybe actually having access to this tree posterior might be useful. So when we we have this posterior distributions of trees, and then we computed credible sets of trees. And then these credible tree sets, we just add the trees with the highest posterior probability until this probability reaches 95%. So this should really account for the trees that are most probable for this particular data. And then we observed that for these data sets, there are a lot of data sets actually where the credible sets contained a huge number of trees, so more than 10,000, for example. And this actually indicates that the data didn't have a lot of signal to favor one tree topology over the other. And so when we then looked actually at the coverage for when we kind of split these data sets, then we can see that when we exclude those data sets which don't have reliable signal, that the coverage of our true tree improves quite a lot. And I think this is really a powerful way to look at this data. Meanwhile, the experimental technology is still a bit noisy, that having access to the tree posterior really allows you to see within a tree, like which parts of the tree are more, we have more signal for, which are more, actually, for which parts we have more certainty and for which parts we don't. Right. And then lastly, we also wanted to infer the cell dynamics from the trees. And to do this, we chose 3 different scenarios. Scenario A is, and just to mention again, so this clock rate, if you just remember, this was the speed of editing, and the edit probabilities is this kind of in the distribution. And so in scenario A, we say, for all of these 10 sites that we have, we have the same clock rate, and we also have the same edit outcome probabilities. In scenario B, we say the clock rate is still shared, but every site can have its own edit outcome probabilities. And then in scenario C, we allow both the clock rate and the edit probabilities to be different for each site. And then, and then we estimate the birth and death rates while fixing the sampling proportion. And this first part here is just to show that we are actually placing our prior on the growth rate and not on these individual rates, because it's easier to parameterize the model in this way. But looking actually at the birth and death rates that we care about, we can observe that scenarios A and B give us very similar estimates. Meanwhile, scenario C gives us a lower cell division and birth and death rate. And when we actually then check back to our experimental setup, the cell division rate that this birth rate corresponds to, so it's a bit maybe not super easy to read off here, but this would be about 0.8 for a time window of 54 hours, gives roughly 5 cell division events, and this is actually matched better by the experimental data than this higher cell division rates that was inferred for the other 2 scenarios. So actually accounting for the edit speed, so allowing the edit speed to vary for the different target arrays was here key to finding the true cell division rate. Right. So a brief outlook. Yeah, something that we think is very important is to improve the runtime for TiDeTree, because this is the a major limitation. And so just to give you a brief idea where our troubles we think mostly come from is these subtrees for which very much consist of identical sequences, and these are very hard to sample and to move around in the tree. And so we are currently thinking along different lines to improve this, so maybe introducing another operator for sampling these subtrees, or also maybe introducing an error model that would allow, that makes, so the basic problem is that some of these trees have 0 likelihoods, they are proposed, and then they are just rejected all the time. And so maybe we're also following up ideas on how we can make these 0 likelihood trees very much non-zero. Exactly. So in summary, TiDeTree estimates timescale trees from lineage tracing data. We think that by having it implemented in these 2, we actually provide users quite a flexibility for selecting different models. So I have shown you today results with just a strict clock, but you could also very much imagine that during, long organism development, this rate of editing might change over time. And for this one, we could readily use relaxed clock models in these 2. And then also for modeling cell type dependent dynamics, we could use multi-type models. And this is all kind of now available very much out of the box. Yes. And then, given that this is a Bayesian framework, we can easily incorporate prior knowledge and assess uncertainty. And I would just quickly highlight some other projects that I'm also currently working on, just in case somebody's interested and wants to chat about them at some other point in time. So I'm also working on like estimating cell dynamics in brain organoids. So yeah, kind of really trying to estimate the differentiation rate between pluripotent to some other intermediate cell stages to neurons from single cell phylogenies. And I'm also currently developing an editing model for the type for some other very cool new single cell lineage tracing technology. And the major improvement here is that the editing duration is a lot longer compared to all other systems that we currently have available. Yes, and so with this, I come to the end of my talk. I'd like to acknowledge everyone involved. That's Tanja Stadler, my supervisor, and who was involved in all of my projects. Tim Bergen, who is involved in the organoid project, together with my collaborators from the Quad Biolab, and then Antoine Swans, who's involved in the typewriter collaboration with the Chanduri Lab. Thank you very much for listening.
Thank you very much. That was a really interesting talk. I apologize for this question, which, of course, comes up. One of the things about these data sets is they can get very, very big. Not like, and I know that you're working on the operators, but in a sense, like that's only going to get you so far. We're talking about maybe 1 million. Is that crazy?
Yeah.
So what's the, is there any prospect for scaling these sorts of things?
I think it really depends on the questions and the application. So I mean, we can also account, we have kind of sampling kind of directly integrated in the model. So one could also think about, yeah, like if we were able to also get meaningful phylodynamic estimates, so really cell developmental process estimates given less data because we explicitly can account for sampling, I think that would be a way to go. I think the other way is maybe once the experimental technologies become less, a bit less noise, or like, I mean, I think there's still some room for improvement on the experimental side. And once we can trust these data sets more, that we don't have to sample the tree posterior, we can then also just, yeah, stick another tree into Beast that is, yeah, where we trust the tree topology and then estimate the phylodynamic parameters based on that fixed tree.
That's interesting.
I think, yeah. So I think these are currently our ways of looking at this. Yeah.
Yeah, can you talk a little bit more? I mean, so let's see, at the very end, you talked about multi-type. So that would be like cell type transitions, right?
Yes, exactly.
I mean, those sorts of I mean, even if you don't have, well, maybe I'll formulate my question a little bit more and ask the question of Sebastian Priot. Thanks, Sophie, for the great talk. I think these Bayesian models are really powerful. One question about scalability, and there's a different scalability question. How do you deal with the fact that there are infinitely many possible indels? And that's thing number one. And then thing number two is, how does the method's runtime scale with the number of indel states?
Right. For infinitely many indel states, I think this wouldn't be a problem because, well, I mean, I think infinitely many, so we currently always restrict ourselves in the data sets that we analyze to the indels we observe, and these are finite. And if we don't observe an indel in our sample data, then from my perspective, I think it's fair to make, yeah, that the probability of observing that indel is then too low, low enough that I think it wouldn't matter if we neglect it. And say for something very large and finite, we can, so then this is mostly affecting our transition probability matrix calculations. And for these we have an analytical formulation. And so this is then constant in the number, so like linear. Sorry, I'm not always kind of perfect with these terms, but this would be then on the order of the number of the different indels that you would expect. Does that answer the question? And I guess, right, and you would still have these matrix, like matrix multiplications then. So this would be then on the order of the index squared. Yeah.
What I was going to ask, and I'd love to get any other questions from the audience, but
yeah.
I mean, it seems like sometimes one of the things that is of interest is not the full tree topology, but what cell types lead to what other cell types. And it seems like one of the nice things that you could do with this Bayesian model is provide an answer to that sort of simplified question, but with error bounds. Is that something that you are currently doing or could do? I mean, it wouldn't have to be a multi-type birth-death process in the sense that, like, I mean, they could all have the same birth-death parameters, for instance.
Yes. Yeah. Absolutely. So yes, yeah, we also think that this would be really nice to characterize exactly what cell type leads to which other, and also what happens if you have some intermediate cell types that are maybe unobserved. I think what we are currently really struggling with is yeah, how many parameters can we concurrently account for. So for example, in this organoid in this brain organoid project, something that's quite hard is estimating all of these parameters from signal that is so sparse. So
At least on my end, we're encountering some technical difficulties. I'm not seeing Sophie anymore, but I think that was a great talk. Thank you for contributing. And we'll be back soon. Thanks, everyone.